Hi class, this is your instructor, Skylar Huff, and we've made it to Chapter 10, Foundations of Genetics. We'll begin with Mendel and the garden pea. So you will contrast the experiments of Gregor Mendel with earlier classic experiments on pea plants. So when you were born, many things about you resembled your mother or father. This tendency for traits to be passed on from parent to offspring is called heredity. So traits are the expressions of a character or a heritable feature. So, how does heredity happen? So, it was before DNA and chromosomes were discovered. This puzzle is one of the greatest mysteries of science. The key to understanding the puzzle of heredity was found in the garden of an Austrian monastery over a century ago by a monk named Gregor Mendel. So, his solution to the crazy puzzle of heredity was the first step in our journey to understanding heredity and one of the greatest intellectual accomplishments in the history of science. So let's get to his experiments. So he was born in 1822 to peasant parents and educated in a monastery. He became a monk and was sent to the University of Vienna to study science and mathematics. Although he aspired to become a scientist and a teacher, he failed his university exams and returned to the monastery where he spent the rest of his life, eventually becoming an abbot. Upon his return, Mendel joined an informal neighborhood science club. Under the patronage of a local nobleman, each member set out to undertake scientific investigations, which were then discussed at meetings and published in the club's own journal. Mendel undertook to repeat some classic crosses with pea plants done in the 1790s. But unlike earlier breeders, he intended to count the numbers of each kind of offspring in the hope that the numbers would give some hint to what was going on. Quantitative approaches to science, measuring and counting, were just becoming fashionable in Europe. So in the experiment, he chose to study the garden pea because of several of its characteristics made it easy to work with. So many varieties were available. He selected 7 pairs of lines that differed in easily distinguishable traits. He knew that the work from early experiments he could expect infrequent version of a character to disappear in one generation and reappear in the next. And he knew, in other words, that he would have something to count. So pea plants are small, easy to grow, and grow large numbers of offspring and mature quickly. And lastly, the reproductive organs of peas are enclosed within their flowers, so left alone, the flowers do not open. They simply fertilize themselves with their own pollen. To carry out a cross, Mendel only had to pry apart the petals, reach in with a pair of scissors, and snip off the male organs, and then he could then dust the female organs with the pollen from another plant to make the cross. So the experimental design was as follows, meaning it was the same as the other experimenters approximately 60 years earlier, only Mendel counted his plants. So the crosses were carried out in 3 steps that are presented in figure 10.3, and you see that on page 169. So he began by letting each variety self-fertilize for several generations. This ensured that each variety was true breeding, meaning that it contained no other varieties of the trait, so it produced only offspring of the same variety within itself being pollinated. So the white flower variety, for example, produced only white flowers and no purple ones in each generation. Mendel called these the parental generation, indicated by capital P. Mendel then conducted his experiment. He crossed 2 pea varieties exhibiting alternative traits, such as a white versus purple. The offspring that resulted in the F1 generation. So finally, he allowed the plants to produce in the crosses of step 2 to self-fertilize. He then counted the numbers of each kind of offspring that resulted in this F2 generation. As reported 60 years earlier and shown in step 3 to your right-hand side, the white flower trait reappears in the F2 generation, although not as frequently as the purple flower trait. So this is that same figure, showing the exact same from number 1 to number 2, what he did, and then the F2 generation. So based on that experiment, let's move right along. What was observed? Mendel experimented with a variety of traits in the garden pea and repeatedly made similar observations. In all, he examined 7 pairs of contrasting traits, all shown in Table 10.1 on page 170. For each pair of contrasting traits that Mendel crossed, he observed the same result, shown in Figure 10.3, which we just saw here. So where a trait disappeared in the F1 generation, only to reappear in the F2 generation, and we'll examine in detail the crosses with flower color. Let's begin with the F1 generation. In the case of flower color, when Mendel crossed purple and white flowers, all the F1 generation plants he observed were purple. He did not see the contrasting trait, white flowers. Mendel called the trait expressed in the F1 plants dominant and the trait not expressed recessive. In this case, purple flower color was dominant to white flower color, which was recessive. Mendel studied several other characters in addition to flower color, and for every pair of contrasting traits Mendel examined, one proved to be dominant and the other recessive. The dominant and recessive traits for each character he studied are indicated in Table 10.1 on page 170. In the F2 generation, after allowing the individual F1 plants to mature and self-fertilize, Mendel collected and planted the seeds from each plant to see what offspring in the F2 generation would look like. He found that some F2 plants exhibited white flowers, the recessive trait. The recessive trait had disappeared in the F1 generation, only to reappear in the F2 generation. It must somehow have been present in the F1 individuals, but not expressed. At this stage, Mendel instituted his radical change in experimental design. He counted the number of each type among F2 offspring. He believed the proportion of the F2 types would provide some clue about the mechanism of heredity. In the cross between purple-flowered F1 plants, he counted a total of 929 F2 individuals. Of these, 705 had purple flowers and 224 had white flowers. Approximately 1/4 of the F2 individuals exhibited the recessive form of the trait. Mendel carried out similar experiments with other traits and found such as round versus wrinkled seeds, and obtained the same results. 3/4 of the F2 individuals exhibited the dominant form of the character, and 1/4 exhibited the recessive form of the character. In words which others, the dominant to recessive ratio among the F2 plants was always approximately a 3-to-1 ratio. And this is Table 10.1. A disguised 1-to-1 ratio. Mendel let the F2 plants self-fertilize for another generation and found that 1/4 were recessive, were true breeding, meaning future generation shows nothing but recessive traits. Thus, the white F2 individuals described previously showed only white flowers in the F3 generation. So you will see that in Figure 10.6. So among the 3/4 of plants that had shown the dominant trait in the F2 generation, only 1/3 of the individuals were true breeding in the F3 generation. The others showed both traits of the F3 generation, and when Mendel counted the numbers, he found that the ratio of the dominant to recessive to again be 3-to-1. From the results, Mendel concluded that the 3-to-1 ratio he observed in the F2 generation was in fact a disguised 1-to-2-to-1 ratio. So 1 true breeding or 1 dominant, 2 not true breeding dominant, and 1 true breeding recessive. 1-to-2-to-1. Mendel proposes a theory, and this is Figure 10.6. To explain the results, he proposed 5 sets of hypotheses that would faithfully predict the results he had observed, now called Mendel's theory of heredity. It has become one of the most famous theories in the history of science. His first: parents do not transmit traits directly to their offspring. Rather, they transmit information about the traits, what Mendel called Merkaba. These factors act earlier in the offspring to produce the trait. Well, these factors act later in the offspring to produce the trait. In modern terminology, we call Mendel's factors genes. His second hypothesis: each parent contains 2 copies of the factor governing each trait. So 2 copies may or may not be the same. If 2 copies of each factor are the same, the individual is said to be homozygous. If the two copies of the factor are different, the individual is said to be heterozygous. The third hypothesis. Alternative forms of a factor lead to alternative traits. Alternative forms of a factor are called alleles. Mendel used lowercase letters to represent recessive alleles and uppercase letters to represent dominant alleles. In modern terms, we call the appearance of an individual its phenotype. Appearance is determined by which alleles of a gene an individual receives from its parents, and we call those particular alleles an individual's genotype. Thus, pea plants might have the phenotype for white flower and the genotype homozygous recessive. The fourth hypothesis. The two alleles that an individual possesses do not affect each other any more than the two letters in the mailbox alter each other's contents. Each allele is passed on unchanged when the individual matures and produces its own gametes. At the same time, Mendel did not know that his factors were carried from parent to offspring on chromosomes. Lastly, the presence of an allele does not ensure that a trait will be expressed in the individual that carries it. In heterozygous individuals, only the dominant allele achieves expression. The recessive allele is present, yet not expressed. These 5 hypotheses, taken together, constitutes Mendel's theory of the hereditary process. Many traits in humans exhibit dominant or recessive inheritance, similar to the traits Mendel studied in peas. So here is just a few of those shown from table 10.2 on page 172. So these are dominant and recessive traits in the human. So given that, let's continue. To analyze the results, it is important to remember that each trait is determined by the inheritance of alleles from parents, one allele from the mother and one allele from the father. These alleles present on chromosomes are distributed to gametes during meiosis. Each gamete receives one copy of each chromosome and therefore one copy of an allele. Consider again Mendel's cross of the purple-flowered and white-flowered plants. Like Mendel, we will assign the symbol dominant P, written in uppercase, to be the dominant allele that is associated with the production of purple flowers, and the symbol recessive P, written in lowercase, to be the recessive allele associated with the production of white flowers. In this system, the genotype of an individual, true breeding for the recessive flowered trait, would be designated as 2 lowercase p's, or homozygous recessive, as both copies of the allele specify the white phenotype. And similarly, the genotype of a true breeding purple flowered individual will be designated as 2 capital P's, known as homozygous dominant, and a heterozygote will be designated as 1 dominant P, 1 recessive P, or 1 uppercase and 1 lowercase, known as heterozygous. Using these conventions and denoting a cross between 2 strains by way of that X, we see on page 173, we can symbolize Mendel's original cross as homozygous recessive by homozygous dominant, shown at the top left. Punnett squares. The possible results from a cross from a true breeding white-flowered plant and a true breeding purple-flowered plant can be visualized with a Punnett square, as shown to you here. In a Punnett square, the possible gametes of one individual are listed along the horizontal side of the square, while the possible gametes of the other individual are listed along the vertical side of the square. The genotypes of potential offsprings are represented by the cells within the square. Please see figure 10.7 now as I show you here on page 173. So that walks you through the setup of a Punnett square, crossing 2 plants that are heterozygous for flower color. Homozygous dominant at the top left as 1 gamete, heterozygous for the 2 diagonals, and then at the bottom right, homozygous recessive. That provides at least for phenotype, or at least let's go back to the genotype frequency. The genotype frequency would be a 1 to 2 to 1 ratio. 1 is homozygous dominant. There are 2 which are heterozygous, and then there is 1 that is homozygous recessive. And as far as Well, I'll save that for just a moment. So the frequency that these genotypes occur in the offspring are usually expressed by a probability, as I just mentioned. For example, in a cross between a homozygous white flowered plant and a homozygous purple flowered plant, heterozygous, or one dominant P and one recessive P, is the only possible genotype for all individuals in the F1 generation as shown before you. This is figure 10.8. So the Punnett square on the left of the figure, 10.8. So because the dominant P is there, is dominant to all recessive P's, all individuals will be purple in the F1 generation. When individuals from the F1 generation are crossed, as shown as a Punnett square on the right-hand side, the probability of obtaining homozygous dominant individuals in the F2 is 25%, meaning the top left only. And then, because 1/4 of the genotypes are homozygous dominant, similarly, the probability of an individual in the F2 generation being homozygous recessive is 25%. So because the heterozygous genotype has two possible ways of occurring, it occurs in half of the cells of the square, giving us the probability of obtaining a heterozygous as being 50%. As I mentioned earlier, you have here, at least for the genotype, a 1:2:1 ratio in that F2 generation. The test cross. So how did Mendel know which of the purple flowered individuals in the F2 generation were homozygous and which were homozygous? It is not possible to simply look at them. For this reason, Mendel devised a simple and powerful procedure called the test cross to determine an individual's actual genetic composition. Consider the purple flowered plant. It is possible to determine such a plant's genotype by simply looking at its genotype. Simply looking at its phenotype, we can not determine the plant's genotype. So again, I say again, the genotype of a plant cannot be determined by simply looking at its phenotype if it is in fact purple. To learn its genotype, you must cross that purple plant with some other plant. So what kind of cross will provide the answer? If you cross it with a homozygous dominant individual, all of the progeny will show the dominant phenotype, whether the test plant is homozygous or heterozygous. It is also difficult to distinguish between the two possible test plant genotypes by crossing with a heterozygous individual. However, if you cross a test plant with a homozygous recessive individual, the two possible test plant genotypes will give totally different results. So, to see how this works, step through a test cross of a purple flowered plant and a white flowered plant. See figure 10.9. This is on page 174. So it shows you the two possible alternatives. Alternative on the left, unknown plant is homozygous dominant. All offspring will have purple flowers, as shown by those 4 purple squares. Alternative 2, which is on the right, unknown plant is heterozygous. One half of the offspring will have white flowers, the other half will have purple flowers, as shown by the 2 white and 2 purple squares. In one of his test crosses, Mendel crossed F1 individuals exhibiting the dominant trait back to the parent homozygous for the recessive trait. He predicted that the dominant and recessive traits would appear in a 1:1 ratio, and that is what he observed. You can see that illustrated in alternative number 2, shown to the right. Test cross using 2 genes. So by using 2 genes, the test crosses can be used to determine the genotype of an individual with the 2 genes which are involved. Mendel carried out many 2-gene test crosses, some of which we will soon discuss. He often uses test cross to verify the genotypes of a particular dominant appearing F2 individual. Thus, the F2 individual showing both dominant traits might have some of the following genotypes as shown. So dominant A, dominant A, dominant B, dominant B, and I'll now say heterozygous A's with homozygous B's, homozygous A's with heterozygous B's, or heterozygous A's and B's. By crossing dominant appearing F2 individuals with homozygous recessive individuals, Mendel was able to determine if either or both of the traits bred true among the progeny and any that did not be heterozygous, and so to determine the genotype of the F2 parent. The laws of Mendel. Mendel's understanding of heredity can be summarized in two simple laws. The first, segregation. Mendel's model brilliantly predicts the results of his test crosses, accounting in a neat and satisfying way the ratios he observed. Similarly, patterns of heredity have since been observed in countless other organisms. So traits exhibiting this pattern of heredity are called Mendelian traits. Because of its overwhelming importance, Mendel's theory is often referred to as Mendel's first law, or the law of segregation. So it states that the two alleles of a trait separate during the formation of gametes, so that half of the gametes will carry one copy and the other half will carry the other copy. Next to the law of independent assortment. Mendel went on to ask if the law of inheritance of one factor, such as flower color, influences the inheritance of other factors, such as plant height. So he followed the inheritance of two separate traits to investigate this question, called a dihybrid cross. He first established a series of true breeding lines of plants that differed from one another with respect to two of the 7 pairs of characteristics, and then crossed them. So you can see this in figure 10.10 on page 175. So it shows an experiment in which the dominant P generation consists of homozygous individuals with round yellow seeds that are crossed with individuals which are homozygous for wrinkled green seeds. This cross produces F1 individuals that have round yellow seeds and are heterozygous for both traits. The chromosomes are then allocated to the gametes during meiosis, such that there are 4 types of gametes for these two traits. You can see that here at the bottom. Mendel then allowed the dihybrid individuals to self-fertilize, and if the segregation of alleles affecting seed shape and alleles affecting seed color was independent. So the probability that the particular pair of seed-shaped alleles would occur together with a particular pair of seed color alleles would simply be a product of the two individual probabilities that each pair would occur separately. For example, the probability of an individual with a wrinkled green seed appearing in the F2 generation will be equal to the probability of an individual with wrinkled seeds. So it's 1 in 4, and that's multiplied by the probability of an individual with green seeds, which is also 1 in 4, or 1 in 16. So in his dihybrid crosses, Mendel found that the frequency of phenotypes in the F2 generation closely matched a 9:3 to 3:1 ratio. And that is predicted by the Punnett square analysis you see in figure 10.10, also shown here now. So he concluded that for the pairs of traits he studied, the inheritance of one trait does not influence the inheritance of another trait, a result often referred to Mendel's second law of inheritance, i.e., the law of independent assortment. So we know now that this result is valid only for genes not located near one another on the same chromosome. So, in modern terms, genes located on different chromosomes are inherited independently of one another. This is the law of independent assortment. So now let's get to how genes influence traits. So it is useful before considering Mendelian genetics to further gain a brief understanding of how genes work. So with this in mind, we will sketch in a broad stroke a picture of how a Mendelian trait is influenced by a particular gene. So we'll use the protein hemoglobin, which is found in human red blood cells, as our example. And you can see figure 10.11 on page 177 to follow along. So hopefully this makes sense. And if it doesn't, please reach out to me at once. From DNA to protein. So each cell in the body of an individual contains a set of DNA molecules called a genome of that individual. The human genome contains about 20,000 to 25,000 genes, and they're parceled into 23 pairs of chromosomes. And you can see that in figure 10.11. So it says that hemoglobin gene, HB, is located on chromosome 11. So individual genes are read from the chromosomal DNA by enzymes that create an RNA transcript of the gene sequence. So after editing out the unnecessary bits, the RNA transcript of hemoglobin leaves the nucleus as messenger RNA and is delivered to ribosomes in the cytoplasm. So each ribosome is a tiny protein assembly plant, and it uses the nucleotide sequence of the mRNA to determine the sequence of amino acids of a particular polypeptide. In the case of beta hemoglobin, the mRNA encodes a strand of 146 amino acids. How proteins determine the phenotype? Let's continue. So what happens is the beta hemoglobin amino acid chain, which resembles beads on a string, shown here in roughly the center, spontaneously folds into a complex three-dimensional shape. This beta hemoglobin associates with other beta chain and two alpha chains to form the active 4 subunit hemoglobin protein, which is shown to the left on page 176. So this is what binds to oxygen in red blood cells. As a general rule, genes influence the phenotype by specifying the type of proteins present in the body, which determines a large measure of how the body works, looks, and function. So how a mutation alters phenotype. So a change in the identity of a single nucleotide within a gene, called a mutation, can have a profound effect on if the new version of the protein folds differently, as this may alter or destroy its function. So for example, how well hemoglobin performs its oxygen transport duties depend on the precise shape of the protein subunits assumed when they fold. So a change in the 6th amino acid of beta hemoglobin from glutamic acid to valine causes the hemoglobin molecule to aggregate into a long chains which deform blood cells into a sickle shape that no longer carry oxygen efficiently. The resulting sickle cell disease can be fatal, as discussed further in section 10.8. Make sure you see section 10.8. So scientists attempting to confirm Mendel's theory often had trouble obtaining the same simple ratios he reported, and often the expression of the genotype was not straightforward. Most phenotypes reflect the action of many genes, and some phenotypes can be affected by alleles that lack complete dominance and are affected by environmental conditions or are expressed together. So continuous variation. Please see your textbook here on page 178. When multiple genes act jointly to influence a character such as height, weight, I would say even skin color, the character often shows a range of small differences. Because all of the genes that play a role in determining the phenotypes, they segregate independently of each other. So we see a gradation in the degree of difference when many individuals are examined. A classic illustration of this sort of variation is seen in figure 10.12 on page 178, a photograph of a college class. The students were placed in rows according to their heights, under 5 feet toward the left and over 6 feet to the right. You can see there is considerable variation in height in this population of students. We call this type of inheritance polygenetic inheritance. Many genes. So we call this gradation in phenotypes continuous variation. In other words, it's not discrete. It's not this or that. Those are discrete characters. So we can describe this variation in a character, such as height of individuals, in figure 10.12. So how can we do that? So individuals range from quite short to very tall, with average heights being more common than either of the extreme. What can we do is this group of variation into categories. So what we often do is group the variation into categories. Each height in inches is a separate phenotypic category. So applying the numbers in each height category produces a histogram, such as that you see in figure 10.12b. The histogram approximately has an idealized bell-shaped curve. The variation can be categorized by the mean and the spread of that curve. So compare this to the inheritance of plant height in Mendel's peas. They were either tall or dwarf. There were no intermediate heights in the plant that existed because only one gene controlled that trait. So that is why I said those are an example of discrete characters, this or that, tall or dwarf. So let's go to pleiotropic effects. So pleiotropic effects, often an individual has an allele that has more than one effect on the phenotype, such as pleiotropic or pleiotropy. So when the pioneering French geneticist Lucien Sinot studied yellow fur in mice, the dominant trait, he was unable to obtain a true breeding yellow strain by crossing individual yellow mice with one another. Individuals homozygous for the yellow allele died because the yellow allele was pleiotropic. One effect was yellow color, but another was lethal developmental defect. So, pleiotropic gene alteration may be dominant with respect to one phenotype, and the consequence would be yellow fur, and a recessive with respect to another could be lethal development. So, in pleiotropy, one gene affects many characters in marked contrast to polygeny, where many genes affect one character. So, pleiotropic effects are difficult to predict because genes that affect the character often perform other functions we may know nothing about. So, pleiotropic effects are characteristic of many inherited disorders such as cystic fibrosis and sickle cell anemia, we'll discuss later in the chapter. In these disorders, multiple symptoms can be tracked or at least traced back to a single gene defect, shown in figure 10.13 and shown to you right there. As shown, cystic fibrosis patients exhibit overly sticky mucus, salty sweat, liver and pancreas failure, and a battery of other symptoms. All are pleiotropic effects of a single gene defect, a mutation in a gene that encodes for a chloride ion transmembrane channel. In sickle cell disease, a defect in the oxygen-carrying hemoglobin molecule causes anemia, heart failure, kidney failure, enlargement of the spleen, and many other symptoms. Incomplete dominance. Not all alternative alleles are fully dominant or fully recessive in heterozygotes. Some pairs of alleles exhibit incomplete dominance and produce a heterozygous phenotype that is intermediate between those parents. For example, if you look at figure 10.14, you will see this on page 179. If you cross a red and white flower Japanese florcock, it'll produce red, pink, and white flowered F2 plants in a 1 to 2 to 1 ratio. The heterozygotes are intermediate in color. There's a mixing effect. This is different than in Mendel's pea plants that didn't exhibit incomplete dominance. Those exhibited complete dominance. So the heterozygotes expressed the dominant phenotype in Mendel's experiments. Epigenetics and environmental effects. And this shows figure 10.14 on page 179. So the degree to which many alleles are expressed depends on the environment. Some alleles are heat sensitive, for example, and traits influenced by such alleles are more sensitive to temperature or light than the products of other alleles. In Arctic foxes, for example, they make fur pigment only when the weather is warm. Can you see why this trait would be an advantage for the fox? Imagine a fox that didn't possess this trait and was white all year round. It would be very visible to predators all summer long, sitting out against the darker surroundings. Yummy. Not for me, of course, but to the predators, excuse me. Sometimes the environment can affect genes directly by adding or removing chemical groups to DNA or the proteins involved in the packaging of DNA into chromosomes, called epigenetic modification. These changes can accumulate as a person ages and can even be passed from one generation to the next. So while most genes are not epigenetically modified, some that influence human behaviors seem to be. So there you have it. Codominance is a gene may have more than 2 alleles in a population, and in fact most genes possess several different alleles. Often, in heterozygotes, there isn't a dominant allele. Instead, the effects of both alleles are expressed. In these cases, the alleles are said to be codominant. Codominance is seen in the color patterning of some animals, such as the roan pattern exhibited by some varieties of cattle and horses. A roan animal expresses both white and colored hairs because both alleles are being expressed at the same time or simultaneously. The horse in figure 10.16 is exhibiting a roan pattern. That's on page 180. It looks like it has gray hairs, but if you're able to examine the coat closely, you will see both white hairs and dark brown hairs. Let's move on now to the ABO blood group. A human gene that exhibits more than one dominant allele is the gene that determines ABO blood type. This gene encodes an enzyme that adds sugar molecules to lipids on the surface of red blood cells. These sugars act as recognition markers for the cells in the immune system and are called surface antigens. The gene that encodes the enzyme designated as I has three common alleles. There is IB, whose product adds galactose. There is IA, whose product adds galactosamine. And recessive I, whose product does not add a sugar at all. So different combinations of the three I gene alleles occur in different individuals, and because each individual person may be homozygous for any allele or heterozygous for any two, an individual that is heterozygous for the IA and the IB alleles produces both forms of the enzyme and adds both galactose and galactosamine to the surfaces of red blood cells. So because both alleles are expressed simultaneously in heterozygotes, the IA and the IB are alleles that are codominant. Both IA and IB are dominant over the recessive I allele because both IA and IB lead to sugar addition and the recessive I does not. So different combinations of the three alleles produce four different phenotypes. So type A blood can come from IA or IAI recessive. Type B blood can come from IB, or IBI recessive. Type AB blood can only come from IB, and lastly, the fourth is going to be recessive I for type O blood. Neither sugar is added, and the recessive I's are homozygotes. So again, you can have homozygous IA for type A, or IAI for recessive. You could have homozygous IB, or IBI, the recessive for type B blood, or you can have IB for type AB blood, or recessive I for type O blood. So these four different cell surface phenotypes are called ABO blood groups. A person's immune system can distinguish between these four phenotypes. If type A individual receives a transfusion from type B blood, the recipient's immune system recognizes the type B blood cells possess a foreign antigen, galactose, and attacks the donated blood cells, causing the cells to clump or agglutinate. So this also happens if the donated blood is type AB blood. However, if the donated blood is type O, it contains no galactose or galactosamine antigens on the surfaces of the blood cells, so it elicits no immune response to these antigens, so it would be no agglutination or no clumping. For this reason, the type O individual is often referred to as being a universal donor, because neither galactose or galactosamine is foreign to type AB individuals, and those individuals are the universal recipients and may receive any type of blood. The human chromosome. Each somatic cell normally has 46 chromosomes, which in meiosis form 23 pairs. Homologous chromosomes can be identified according to the size, shape, and appearance. Of the 23 pairs of human chromosomes, 22 are perfectly matched in both males and females, which are called autosomes. Numbers 1 through 22 class are autosomes. The 23rd pair are the sex chromosomes, consisting of 2 similar chromosomes in females and 2 dissimilar chromosomes in males. So females have homozygous chromosomes, males have heterozygous chromosomes, i.e. X and Y, as opposed to the former being XX for females. The genes present on the Y chromosome determine maleness. Therefore, humans who inherit the Y chromosome develop into males. Sometimes during meiosis, homologous chromosomes or sister chromatids that paired up during metaphase remain stuck together instead of separating. The failure of chromosomes to separate correctly during either meiosis 1 or meiosis 2 is known as non-disjunction. Non-disjunction leads to aneuploidy, being off by one chromosome or off by a number of chromosomes. So otherwise referred to an abnormal number of chromosomes. So the non-disjunction you see in figure 10.18 on the top right-hand corner of page 181 occurs because of the homologous pair of larger chromosomes fail to separate in anaphase 1. The gametes that result from this division have an unequal number of chromosomes in each. So under normal meiosis, all gametes would be expected to have 2 chromosomes. But as you see here, 2 of these have 3 chromosomes, which will be N plus 1, and the others have just one, which would be N minus 1 and N minus 1, the definition of aneuploidy. Almost all humans of the same sex have the same karyotype, simply because the arrangements don't work out well. So, humans who have lost even one copy of an autosome, called monosomic, do not survive development. In all but a few cases, humans who have gained an extra chromosome, an extra autosome, called trisomics, do not survive. However, five of the smallest chromosomes, those numbered 13, 15, 18, 21, and 22, can be present in humans as 3 copies and still allows the individual to survive for a time. The presence of an extra chromosome 13, 15, or 18 causes severe developmental effects, and infants with a genetic makeup die within a few months. In contrast, individuals who have an extra copy of chromosome 21, or more rarely chromosome 22, usually survive to adulthood. In such individuals, the maturation of the skeletal system is delayed, so they generally are short and have poor muscle tone. Their mental development is also affected, and children with trisomy 21 or 22 are always mentally impaired. And this is from your textbook, not Huff. So this shows non-disjunction. Now on to Down syndrome. The developmental defect produced by trisomy 21, an extra copy of the 21st chromosome, as seen in the karyotype 10.19 on page 181, was first described in 1866 by J. Langdon Down. For this reason, it is called Down syndrome. About 1 in every 750 children exhibits Down syndrome, and the frequency is similar in all racial groups. It is more common in children of older mothers. So the graph shown in figure 10.20, as you see here, shows an increasing incidence in older mothers. So in mothers under 30 years old, the incidence is only 0.6 per 1,000, or 1 in every 1,500 births. Whereas mothers ages 30 to 35 years old, the incidence doubles to about 1.3 per 1,000 births, or 1 in every 750 births. In mothers over 45, the risk is as high as 63 per 1,000 births, or 1 in 16 births. So the reason that older mothers are more prone to Down syndrome babies is that all eggs that a woman will ever produce are present in her ovaries by the time she is born. And as she gets older, they may accumulate damage that can result in non-disjunction. Non-disjunction, as noted, there are 22 of the 23 pairs of human chromosomes that are perfectly matched in both males and females, and they're called autosomes. So the remaining pair are the sex chromosomes, the X and Y. In humans, as in Drosophila, females are XX and males are XY. This is your textbook. So any individual with at least one Y chromosome is male. This is in your textbook. The Y chromosome is highly condensed and bears few functional genes in most organisms. Some of the active genes on the Y chromosome does possess and are responsible for features associated with maleness. So individuals who gain or lose a sex chromosome do not generally experience severe developmental or abnormalities caused by changes in autosomes. Such individuals may reach maturity, but somewhat abnormal features. So non-disjunction of the X chromosome, and that's when it fails to separate during meiosis, and some gametes possess both X chromosomes, so they are XX gametes. The other gametes that result may have no sex chromosome and are designated as O, or 0. So figure 10.21 shows what happens if gametes from the X chromosome non-disjunction combine with sperm. So if an XX egg combines with an X sperm, the resulting XXX zygote develops into a female who is taller than average, but other symptoms can vary greatly. Some are normal in most respects. Others have lower reading and verbal skills, and still others are mentally I won't have what your textbook says there, but I'll say they are mentally impaired. I will not say what your textbook has typed there on page 183. So if an XX egg combines with a Y sperm, the XXY zygote develops into a sterile male who has many female body characteristics and in some cases diminished mental capacity. This condition is called Klinefelter syndrome and occurs in about 1 in 500 male births. If an zero egg or an O egg fuses with a Y sperm, OY zygote is non-viable and fails to develop further because humans cannot survive if they lack the genes on the X chromosome. If a O or a zero egg fuses with an X sperm, the XO develops into a sterile female of short stature with a webbed neck and immature sex organs that do not undergo changes during puberty. The mental abilities of XO individuals are normal in verbal learning, but lower in nonverbal math-based problem solving. This condition is referred to as Turner syndrome, and it occurs once in every 5,000 female births. So non-disjunction of the Y chromosome is next. The Y chromosome can also fail to separate in meiosis, leading to the formation of YY sperm. When these sperm combine with X eggs, the XYY zygotes develop into fertile males of normal appearance. The frequency of XYY is about 1 in every 1,000 newborn males. Mutations. The proteins encoded by most of your genes must function in a very precise fashion for you to develop properly and for many of the complex processes of your body to function correctly. Unfortunately, genes sometimes sustain damage or are copied incorrectly. We call these accidental changes in gene mutations. So, mutations occur only rarely because your cells monitor your genes and attempt to correct any damage they encounter. Still, some mutations get through. Many of them are bad for you and in one way or another. So it is easy to see why mutations hit genes at random and imagine that you randomly change the number or part of the design of a jet fighter. Something won't matter. Sure, sometimes it won't matter critically. A seatbelt becomes a radio, you say. But what say a key rivet in the wing becomes a roll of toilet paper? Well, the chance of a random mutation in a gene improving the performance of a protein is about the same as that of the randomly selected part making the jet fighter fly faster. So most mutations are rare in human populations. Almost all result in recessive alleles, and so they are not eliminated from the population by evolutionary forces because they are not expressed in most individuals in which they occur. So do you see why they occur mostly in heterozygotes? Because the mutant alleles are rare, it is unlikely that a person carrying a copy of the mutant allele will marry someone who also carries it, and instead he or she will typically marry someone homozygous dominant. And so none of their children would be homozygous for the mutant allele. While most mutations are harmful to normal functions and usually recessive, that is not to say that all mutations are undesirable. Some mutations can lead to enhanced function. So nor are all mutations recessive, and rarely they can occur in dominant alleles. Some cases, particularly mutant alleles, may have become more common in human populations. So in these cases, the harmful effects that they produce are called genetic disorders. So the most common genetic disorders are listed in Table 10.3 on page 184. To study heredity, scientists look at the results of crosses that have already been made. They study family trees or pedigrees to identify which relatives exhibit a trait. They can often determine whether the expression of a gene is dominant or recessive and whether the gene produced is sex-linked, that is, located on the X chromosome or autosomal, i.e., on an autosome. Frequently, the pedigree will also help an investigator infer which individuals in a family are homozygous and which are heterozygous for the allele specifying the trait. In the pedigree, females are indicated with circles and males with squares, the lines that connect parents and display the offspring. Solid shapes indicate an individual is affected by a disorder, half-filled shapes indicate a carrier, someone who is heterozygous, and open shapes indicate an individual is not affected. Hemophilia, an X-linked or sex-linked trait. Blood in a cut clots as a result of polymerization of protein fibers circulating in the blood. A dozen proteins are involved in this process. and all must function properly for a blood clot to form. So a mutation causing any of these proteins to lose their activity leads to a form of hemophilia, a hereditary condition in which blood clots slowly or not at all. Hemophilias are recessive disorders expressed only when an individual does not possess any copy of the normal allele and so cannot produce one of the proteins necessary for clotting. So this is that generalized pedigree mentioned earlier to you all. Circle, female, square, male, half circle, carrier, enclosed completely, affected individual. And this is table 10.3. So most of the genes that encode blood clotting proteins are on autosomes, but 2 are on the X chromosome. These 2 genes are thus sex-linked. Any male who inherits a mutant allele will develop hemophilia because his other sex chromosome, the Y chromosome, that lacks any alleles of those genes. So the most famous instance of hemophilia, often called the royal hemophilia, is sex-linked form that arose in the royal family of England. So this hemophilia was caused by mutation in gene number 9 that occurred in one of the parents of Queen Victoria of England. So the pedigree, shown in figure 10.24 on page 186, shows that in 6 generations, Queen Victoria, 10 of her male descendants, all had hemophilia, the solid squares. So the present British royal family has escaped this disorder because Queen Victoria's son, King Edward VII, did not inherit the defective allele, and all the subsequent rulers of England are his descendants. So 3 of Victoria's 9 children did receive the defective allele. However, they carried it by marriage into many of the other royal families of Europe. As you will learn in chapter 13, this type of hemophilia has been successfully treated by using gene therapy. Sickle cell disease. It is a recessive hereditary disorder, and its inheritance is shown in the pedigree, shown on page 10.25. Shown in figure 10.25 on page 187. So it shows in which affected individuals are homozygous carrying 2 copies of the mutated gene. So affected individuals have defective molecules of hemoglobin, so the protein within red blood cells that carries oxygen. Consequently, these individuals are unable to properly transport oxygen to their tissues. So the hemoglobin in the defective red blood cells differs from that of the normal red blood cells in only one of the beta hemoglobin's 146 amino acid subunits. In the defective hemoglobin, the amino acid valine replaces glutamic acid at the single position of each of the 2 beta subunit proteins. Interestingly, the position of the change is far from the active site of hemoglobin, where the iron-bearing heme group binds oxygen. Instead, the change occurs on the outer corners of the 4 subunit hemoglobin molecule. So the reason why this is so catastrophic is that the sickle cell mutation puts the very nonpolar amino acid valine on the surface of the hemoglobin protein, creating a sticky patch that sticks to other such patches on nonpolar amino acids that tend to associate with one another in polar environments such as water. The defective hemoglobin molecules adhere to one another in chains. So these hemoglobin chains form stiff rod-like structures that result in a sickle-shaped red blood cell. So as a result of their stiffness and irregular shape, these cells have difficulty moving through the smallest blood vessels, and they tend to accumulate in these vessels and form clots. So people who have large proportion of sickle-shaped red blood cells tend to have intermittent illness and a shortened lifespan. Resistance to malaria, however, does come from such. So individuals who are heterozygous for the sickle cell allele are generally indistinguishable from the normal persons. However, some of the red blood cells showing sickling characteristic when they are exposed to low levels of oxygen. The alleles are responsible for sickle cell disease is particularly common among people of African descent because the sickle cell allele is more common in Africa. About 9% of African Americans are heterozygous for this allele, and about 0.2% are homozygous and therefore have the disorder. In some groups of people in Africa, up to 45% of all individuals are heterozygous for this allele, and fully 6% are homozygous and express the disorder. So what factors determine the high frequency of sickle cell disease in Africa? It turns out that heterozygosity for sickle cell anemia increases resistance to malaria, a common and serious disease in Central Africa. Comparing the two maps shown in figure 10.26, you can see that the area for the sickle cell trait matches very well with the incidence of malaria. So the interactions of sickle cell disease and malaria will further be discussed in the 14th chapter of your text. Class, this has been what I call your instructor, Skylar Huff, and this has been Chapter 10, Foundations of Genetics. Please be sure to take time to go through this chapter with a fine-toothed comb. And in that, be sure to put the chapter to work on page 188 and retrace your learning path. Prepare well for your test because there will be some problems to work out. Thank you all for listening.